Prokaryotes, bacteria and archaea, reproduce asexually through cell division into daughter cells that carry on the genetic sequence into the next generation. Some prokaryote bacteria do conjugate to exchange DNA. However, the way bacteria exchange DNA is not true sexual reproduction. True sexual reproduction is synonymous with eukaryotes, animals, plants, fungi, and single-celled protozoans. With a few exceptions, all eukaryotes reproduce through sexual reproduction, the deliberate production of haploid gametes, sperm and eggs, through the DNA replication process called meiosis. The DNA chromosomes of these sex gametes join together, the sperm fertilizes the egg, and the offspring cells have chromosome pairs, one chromosome from each parent. The offspring will have the characteristics of both parents. Although the expression of these characteristics might not be equal depending on environmental factors, both parents are represented equally in the DNA of the chromosome pairs of the offspring. It is thought that sex was invented by the first eukaryote cells, nearly 2 billion years ago. There are two types of true sex, dioicous sex and hermaphroditic sex. Dioicous sex means there are male and female versions of the same species. Hermaphroditic sex means each organism can produce both male and female gametes. In the absence of a partner, hermaphrodites can fertilize their own eggs. Hermaphroditic sex is limited to some self-pollinating plants and a few primitive worms. Male and female, and been the dominant form of sex for multicellular creatures for 500 million years. It is still a mystery why dioecious sex persisted in eukaryotes. There is no definite advantage of sexual reproduction over asexual reproduction with conjugation. Only half the population can birth new members, and only half the genes of any organism will be passed on through sexual reproduction. Asexual reproduced beings can weed out deleterious mutations and develop genetic variants that respond to new environments as quickly, if not more quickly, than sexually reproduced beings. A whole panoply of hypotheses has been proposed as to why dioecious sex has persisted. Most of these hypotheses lack definite proof and are subject of dispute. Many books could be written on the advantages and disadvantages of sexual reproduction. The real reason for sex is still a scientific mystery. I will only give a few advantages of sexual reproduction. These include DNA repair and species conservation. Whenever there is gamete production, there is DNA repair that occurs during the copying phase of meiosis. As for species conservation, Gorelick and Heng propose that sexual reproduction puts an upper limit on the genetic variation between generations. Major mutations are rejected during meiosis. Sex between closely related species with different numbers of chromosomes will produce no fertile offspring, and so maintain the separation between species. Small variations within a species are encouraged. Minor deleterious genes are suppressed or eliminated. Beneficial mutations can be suppressed for many generations only to appear during a period of environmental stress when the beneficial mutations become important. In sexual reproduction, the goal of DNA replication is the production of haploid cells that can be used as gametes, the egg and sperm cells of eukaryotes that practice sexual reproduction. As the meiosis to produce haploid cells repeats many of the steps of diploid cell mitosis, it may be worth giving a brief review of mitosis of diploid cells. In diploid cell production, the cell undergoes mitosis. Before mitosis can begin, the DNA must replicate itself, so there are 2 copies of every chromosome. There are 5 phases of mitosis: prophase, metaphase, anaphase, telophase, all followed by cleavage and cytokinesis. In prophase, the 2 copies of the chromosomes join to form the familiar X shape we recognize as a homologous chromosome pair. The nuclear envelope dissolves. The centrioles take up positions on the opposite side of the cell. The spindle fibers grow from each centriole. The chromosome pairs line up on the equator of the spindle. B for In metaphase, the spindle fibers pull the chromosomes apart so that each daughter cell will get one copy of the chromosome. In anaphase, the daughter chromosomes are pulled by the spindle fibers towards the centrioles. In telophase, the nuclear envelopes reform around each set of daughter chromosomes. The chromosomes begin to unwind back into amorphous chromatin. In cytokinesis, the outer cell membrane pinches and cleaves, and the daughter cells move away from each other as separate diploid cells. Mitosis and meiosis are cell division processes exclusive to eukaryote cells. Prokaryotes, bacteria and archaea, do not undergo mitosis or meiosis. Prokaryotes don't have a nucleus. Their DNA is shaped into rings that float freely in prokaryote cytoplasm. When the prokaryote has grown enough material to multiply, the prokaryote cell simply replicates its DNA and then distributes that DNA equally between the daughter cells during cell division. It is a mystery how eukaryotes develop the apparatus, cytoskeleton, centrioles, and microtubules that make up the spindle fibers and other structures and procedures that carry out the mitosis and meiosis processes. How mitosis developed in eukaryotes is a limit on our knowledge. Prokaryotes developed 3.5 to 4 billion years ago. The first eukaryotes appeared on Earth sometime around 2 billion years ago. There are no living species between prokaryotes and eukaryotes. Soft microbes don't fossilize well, so there is little to no physical evidence of missing links between eukaryotes and prokaryotes that are preserved from 2 billion years ago, when eukaryotes might have been transitioning from prokaryotes. There is the endosymbiotic theory put forth by Lynn Margulis, that eukaryotes evolved by engulfing prokaryotes and made them into organelles like mitochondria and chloroplasts by endosymbiosis. How mitosis developed is a mystery that may never be solved. Meiosis has many of the steps of mitosis, except those steps are repeated twice and with a few extra steps. As with mitosis, meiosis starts with a set of pairs of chromosomes. To keep things simple, let's focus on a single pair of chromosomes, with the thought in mind that multicellular creatures have a set number of multiple chromosome pairs. Humans have 23 pairs of chromosomes. Other species have a different number of chromosome pairs. The figure shows an overview of meiosis from one parent cell through DNA crossover to 4 haploid daughter cells. The figure demonstrates how crossover redistributes DNA by mixing the parent strands. At this point, we could continue a meiosis with no crossover as shown on the left side of the figure. However, as the point of sexual reproduction is to mix and recombine the genes, we will take the crossover pathway of meiosis as shown on the right side of the figure. Meiosis is a double cell division process called meiosis I and meiosis II. If we take a good look at the figure, we see an overview of meiosis from one parent cell with crossover and no crossover. The figure demonstrates how crossover redistributes DNA by mixing the parent strand. With no crossover on the left side, the paternal and maternal DNA are 100% preserved. The offspring would be effectively a half-sister of the parent. With crossover, there is more than 50% more mixing, with only a 50% chance the offspring would be a half-sister or half-brother of the parent. Each pair is made up of paternal chromosomes and maternal chromosomes, reflecting the fact that we get one chromatid from our father and one chromatid from our mother as shown in the figure. Before meiosis can begin, each chromatid undergoes DNA replication so that the result is homologous chromosome pairs, as shown in the figure. Homologous means that they are identical in that each chromatid in a chromosome pair has an identical DNA sequence reflecting the original paternal and maternal chromosome. The cell in the figure is a tetrad or tetraploid cell with 4 chromatids or 4 complete gene sequences. Geneticists often label each complete gene as N. So, a normal diploid cell has 2 N gene sequences. A tetrad or tetraploid has 4 N gene sequences. As we shall see, by the end of meiosis, the 4N tetraploid will be reduced to a single gene sequence or a 1N haploid cell. In the next set of figures, note the aqua-colored gene on the paternal chromosome legs and the pink-colored gene on the maternal-colored legs. The aqua genes are on the paternal chromosomes and the pink genes are on the maternal chromosomes. The first true step of meiosis I is prophase I. In prophase I, the replicated homologous pairs form the familiar X shape of chromosome pairs. Here, prophase I of meiosis I deviates from the prophase of mitosis by a synapsis step followed by crossover, the swapping of genes between chromatids. In synapsis, the arms or legs of the paternal and maternal overlap and form a four-legged tetrad structure shown in the figure. During synapsis, DNA is swapped between the legs of the chromosomes. This gene swapping is known as crossover. Synapsis and crossover are unique to meiosis. It is the only time in eukaryotic cells that genes can be transferred from one DNA strand to another. Crossover is bidirectional, meaning the matching gene sequences from each chromatid leg are swapped simultaneously. The swapped gene sequence are identical in length but may have genes with different combinations of letters A, G, C, T. The letter combinations are unique to the parents and grandparents from which they were passed down. Crossover and recombination allow sections of each chromatid to contain DNA sequences from both parents, so that the offspring of the two parents will contain some of the gene sequence of all four grandparents. In the figure, we can see that the aqua-colored gene from the paternal chromosome has swapped places with the pink-colored gene from the maternal chromosome. Once crossover and DNA recombination is complete, the other steps of prophase continue as in mitosis. The centrioles take their places at opposite ends of the cell. The spindle fibers extend across from centriole to centriole. The chromosomes line up on the equator of the spindle fibers. Another difference between meiosis and mitosis occurs at the beginning of metaphase. Instead of pulling the X chromosome pairs apart as is in mitosis, in meiosis, the mixed X pairs are pulled whole towards each centriole through metaphase, anaphase, and telophase. The result is, by the end of cytokinesis and cell division, the two diploid cells formed have very different chromosomes in their nuclei. As we can see in the figure, one diploid cell has most of a mixed paternal chromosome, with a pink maternal gene at the bottom of one leg. The other diploid cell has most of a mixed maternal chromosome with an aqua paternal gene at the bottom of one leg. For illustration purposes, I deliberately restricted the mixing to one pair of genes. In the real case, the crossover and mixing of paternal and maternal genes would have been much more extensive. Meiosis I has reduced the N number of gene sequences from 4N down to 2N in each cell. At the end of meiosis I, in the cytokinesis or cell division stage, the mixed chromosomes are pulled apart and distributed into two diploid cells that are genetically different from the parent cell. The end of the first cytokinesis marks the end of meiosis I and the beginning of meiosis II. In prophase I through metaphase II, anaphase II, and telophase II, the X-form chromosomes line up on the centriole spindles, and the individual chromatids from each X-form chromosome are pulled apart. The results of the cytokinetic division II of the two diploid cells is 4 haploid cells, each containing one chromatid or a unique single-strand chromosome that are mixed-up versions of the paternal and maternal chromosomes of the original parent cell. Meiosis I reduces the N number of gene sequences from 4N down to 2N in each cell. Meiosis II reduces the N number of gene sequences from 2N down to 1N in each cell. These haploid cells become the gametes or the egg and sperm cells that are exchanged between sexual partners. One last difference between male and female gametes. In males, all 4 gametes become sperm cells. In females, only one of these haploid cells will contain all the mitochondria and organelles of the egg. The other 3 haploid cells will become polar bodies that will be sacrificed as food for the growing zygote should the egg be fertilized by a passing sperm cell. By the end of meiosis II, the 2 diploid cells have each undergone a second cell division. The second cell division distributes the 4 unpaired chromatids among 4 haploid cells. These will become the sex gametes, that could be the sperm or egg DNA that will form the next generation of offspring. We can see the offspring will get a mix of DNA from all 4 grandparents. When 2 haploid gametes come together, the result is a normal diploid cell. 2 1N haploid gene sequences become the 2 N gene sequences of a normal diploid cell. In other words, when haploid sperm fertilizes a haploid egg, the result is a diploid zygote. A zygote is the unicellular stage of what will become a multicelled organism. In human development, within 30 hours of fertilization, the zygote undergoes its first mitosis to become a 2-cell. By the 4th day of conception, the dividing mass has 16 cells and is called a morula. By day 5, the morula begins to hollow out and becomes a blastocyte. By day 7, the blastocyte implants in the uterine wall. Upon implantation, the blastocyte is renamed as an embryo. The embryo will undergo mitosis many times before its cells begin to differentiate into organ cells, skeletal cells, muscle cells, and brain cells that make up an embryo, then a fetus, then a baby. Around week 10, the embryo will begin to look human enough to be called a fetus. A human mother will call the growing being in her uterus a baby from the moment she becomes aware that she is pregnant.
And that is my take on sex, meiosis, and haploid cell formation, and how these processes lead to the formation of a fertilized egg and then a baby. In my next video, I will get into detail on how RNA is transcribed from DNA. Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.